First of all, I'd like to welcome everyone to the start of this second day of the symposium on genetic medicine, evolution, revolution. Next slide, please. Just a reminder to everyone, if you want to ask a question, there are two ways of asking questions. On the tab below, you can type in the question on the Q&A button, or you can raise your hand. We have a moderator, Shell, who will coordinate asking questions to our speakers. Speakers have 20 minutes, followed by 10 minutes of questions. So today we have three very illustrious speakers. We're going to start off with Jeff Ulmer, followed by Norbert Pardy, and we're going to finish off with Peter Collis. Jeff is going to talk us through a little bit about nucleic acid vaccines and particularly RNA vaccines. Norbert will take us through his academic research on the use of base modifications in mRNA vaccines. And Peter's going to show us how LNPs are being used for more than just RNA vaccines. So next slide. That's the first session. Second session, we're going to have, it's going to be coordinated by Padma, our Senior VP of Business Development, talking about partnering and building genetic medicines of the future. And there we have Kurt Kamrud from Fever Animal Health, Robin Shattuck from Imperial College London, and Sharon Wang from Ultragenyx Pharmaceuticals. And then we're going to close out the session with Michael Houghton, who's going to give us his perspectives on RNA vaccines, a field that he's very excited to be probably entering in the near future. So we'll then have a follow-up question session at 12:30 Eastern time with the panel listed there. So if there are any questions that we don't get to during the speaker sessions, we'll try and address them in this additional 30-minute session. And then we'll have the closing ceremony where Precision Nanosystems will celebrate its 10th year in the industry. So with that, we're going to transition to Jeff Ulmer, our first speaker. I've known Jeff for a very long time. I worked with him at Novartis and briefly at GSK. He's definitely a pioneer in the nucleic acid vaccine field. He's worked at Merck, Chiron, Novartis, and GSK. He's one of the few people that's followed the nucleic acid vaccine from the very beginning. He started at Merck with DNA. He continued that journey in Chiron. He transitioned to the viral replicon particle there, where they were delivering RNA with a viral particle. Chiron became Novartis Vaccines in 2006, and he was transitioned then to delivering self-amplifying RNA rather than virally with lipid nanoparticles. That's when I joined the team at Novartis. And then he became site head in the US during that transition of the program to GSK and its migration down into Maryland. And he oversaw work at GSK, various programs, until he recently retired a month ago. And now he, in his retirement, is consulting in the vaccines field. So with that, Jeff, I'll hand it over to you, and it'd be great to hear you talk.
Well, thank you very much, Andy. Suddenly I feel old, but thank you for the kind introduction. It's my honor and pleasure to be part of this symposium. Let me begin by sharing my screen, and let me know when you see it.
We see you, you're great.
Excellent. Well, thanks again, Andy. I think for vaccines, undoubtedly one of our most effective medical intervention, having virtually eliminated many different infectious diseases. Yet there are many challenges that remain, and they're listed here, including existing vaccines that are suboptimal, whether it be from lack of potency in the entire population, for example, tuberculosis, or whether it's the use of antiquated methods in the production of the vaccine, such as influenza. Second, and very importantly, there remain many major unmet medical needs for which vaccines could have a major impact, beneficial impact, but have not yet been developed. HIV, respiratory syncytial virus, and cancer are just a few. Vaccines R&D process overall is very lengthy, complicated, and costly, and there are certainly opportunities for us to streamline this. And finally, we have been woefully inadequate in preparing for emerging outbreaks of infectious diseases until recently. Some very exciting new data that I'll talk about. To understand a little bit better where there may be opportunities to intervene to address these issues, I think it's worthwhile to take a look at what we currently have in our toolbox of technologies for vaccines. Among the most effective of our vaccines, and the first to be developed really, were the live attenuated organisms, which have been engineered in a way to maintain their infectivity but render them safe. And in doing so, they launch a live infection and thereby induce broad and potent immunity, including both antibodies and T cells, including cytotoxic T lymphocytes. And as such, are very effective to protect from infectious disease. However, there are safety risks with this type of approach. Reversion to virulence is always a possibility, and they're not always feasible to make. Not all types of viruses are amenable to this live attenuated approach. A safer approach and a simpler approach is to take the pathogen and inactivate it in some way, usually through chemical means, to reduce the safety risk. However, in doing so, it narrows the type of immunity that is induced, typically antibody-mediated protection. And the process to inactivate sometimes can affect the integrity of the antigen that's on the virus. And because it contains the entire organism, it often has antigens that are either unnecessary or even potentially counterproductive to protective immunity. And like live attenuated viruses, it's not always feasible. Not all viruses are amenable to growth in cell culture. And this really led to subunit vaccines, either through purifying the most important elements, whether they be proteins or polysaccharides, or when recombinant DNA technology became available to produce recombinant protein antigens. And this increased safety provides a more defined composition and importantly focuses the immunity on the key antigens and eliminates those that can be red herrings. But it also induces more narrow immunity, typically limited to antibody-mediated protection. The proteins themselves are not particularly immunogenic, so they often require adjuvants. And because it requires cell culture to produce these proteins, the R&D timelines are long and complicated. And hence this provided the impetus for genetic medicines, nucleic acid-based vaccines to ideally combine all of these positive attributes while avoiding the limitations of these existing vaccines. The types of genetic medicines are shown here, and they include recombinant viral vector systems, one that's commonly used and being used for COVID-19 is based on adenovirus. And in this case, the viral vectors are modified to retain to a certain extent their infectivity, but render them safe. You insert the gene of interest into the genome of the vector, and in doing so, when these vectors are delivered, you express the antigen in a way similar to a virus infection. A simpler approach is to use the nucleic acids themselves, whether it's a plasma DNA produced in E. coli, into which you've cloned the gene of interest, or more recently, messenger RNA, and there are three basic flavors of mRNA vaccines. Conventional mRNAs that have the gene of interest flanked by non-coding regions on the 3' and 5' end that either contain the natural bases or use modified bases, and we'll hear a lot more detail about that a little bit later. And the third and fundamentally different approach is to use what's called self-amplifying RNA, where an RNA viral genome has been modified by removing the structural genes and replacing them with the gene of interest. And in this way, when the RNA is delivered into the cytoplasm of a cell, it uses the host cell protein synthesis machinery to launch the expression of the RNA replicase machinery, which makes copies of the RNA. And downstream subgenomic promoter produces the antigen of interest. And in this way, potentially tens of thousands of copies of the RNA replicon can be produced from a single copy that's delivered into the cell. There are four key pillars for all of these genetic medicines. The antigen, of course, is critical to focus the immune response on not only the right antigen, but the right form of the antigen. And this is particularly true for viral targets where three-dimensional conformational epitopes are often the targets of protective antibody responses. I think a good recent example of how engineering of an antigen can make a big difference is with the respiratory syncytial virus F protein, which in the virus exists in two very different conformations, a pre-fusion state, which is on the surface of the virus before it infects the cell, and a post-fusion state, which it flips into upon entry into the cell. And recent work by the Vaccines Research Center, Peter Kwong's group and Jason McClellan, who identified the structural elements of the RSV antigen and engineered it to be expressed as a more stable form of the pre-fusion, made a big difference in the potency of the vaccine. And think this is going to be generally true for viral targets. The vectors, of course, are important, whether it's a viral vector, a DNA, or an RNA, or a self-amplifying RNA. These, the choice of these vectors may actually be determined by the particular target, remains to be seen, but it's useful to have a number of different vectors in the toolbox in the early stages of discovery to elucidate which is going to be the most appropriate vector system for your particular target. Delivery, of course, is important. The viral vectors have a built-in efficient delivery system with the viral infectivity of the viral particle itself, but the nucleic acids, DNA and RNA, require help. And this can come in either the form of a physical delivery system like electroporation, or more commonly through formulations to use nanoparticle-based approaches to not only protect the nucleic acid from degradation, but also to facilitate delivery into the cell, whether it be in the cytoplasm in the case of an RNA or the nucleus in the case of a DNA. And then finally, one of the key differentiators of genetic medicines is the way they're manufactured, and I'll get into the details a little bit later. The field itself is relatively young, only a little bit more than 30 years old, where back in the mid-'80s, viral vectors were first used for gene therapy approaches. In this case, it was a retroviral vector. Since then, a wide variety of different vector systems have been used for both infectious and non-infectious diseases. In 1990, an observation by John Wolfe and his colleagues showing that simple injection of nucleic acids, both plasmid DNA and messenger RNA, could result in the transfer of those genes into cells and the production of the protein, really launched the field of nucleic acid vaccines. A few years later, while I was at Merck, we demonstrated proof of principle for the DNA vaccine approach, showing that an influenza DNA vaccine could confer protective immunity in an animal model. And concurrently, Pierre Molien and his team showed that an mRNA vaccine expressing an influenza gene could induce immune responses. A few years later, the first DNA vaccine clinical trials began with our flu vaccine at Merck, as well as therapeutic HIV vaccines developed by David Weiner at Penn and Britta Warren at the Karolinska Institute. It was about 10 years later that the first cancer RNA vaccine trials began in Germany, I believe. And then most recently, we've seen how the technology can really facilitate a rapid response.
I just want to check. I think your sound got a little bit lower.
Okay,
So I just
is
wanna
that
make sure we're
is
okay.
that better? I can speak closer to the microphone. Is that better?
Thank you. Sorry to interrupt you.
No problem. And so recently we've seen that the technology is amenable to rapid response, both in the DNA vaccine format, where Inovio responded to Zika vaccine by going from sequence to phase 1 clinical trial in just a few months. And then, of course, this year we've had a spate of genetic medicine Covid19 vaccine trials begin, and I'll get into some details a little bit later. But first, let's go back to the challenges that we're seeing with vaccines. And making better and new vaccines, I think, is a big opportunity here for the use of genetic medicines and genetic vaccines to address this issue. I think to best illustrate that is to show this table that we put together back at Novartis, showing the broad utility of the self-amplifying messenger RNA vaccine in a wide variety of animal models, mostly viral targets, but also parasite targets like malaria, bacterial targets like tuberculosis, Group E, Group A streptococcus, and cancer targets in a wide variety of different species, including non-human primates. And notably, we benchmarked with licensed human vaccines in the rabies setting as well as the flu setting, and showed that the RNA vaccine was at least as potent as a full human dose of these commercially available licensed human vaccines that have known to be potent and effective in humans, which I think bodes quite well for the technology. Similar lists exist for DNA vaccines and viral vector vaccines, but safe to say, at least in animal models, the data suggest that there's very broad utility for this type of an approach. With regard to streamlining the vaccines R&D process, vaccines typically take a very long time to produce. They're complicated and costly processes to develop and manufacture them, often taking several decades. And indeed, if you look at the various vaccines that are in use today, many of them took many, many decades from the time the pathogen was identified to the introduction of the vaccine. The key difference between these vaccines and the genetic vaccines that I've been talking about is the way they're made, in particular for the messenger RNA vaccines, which are produced completely synthetically. They don't require cell culture. They're very simple purification processes, and they don't require a big footprint or novel equipment. And because the drug substance itself is basically the same from one product to the next, with the difference being really only the coding sequence of the gene insert, it can be treated pretty much the same from product to product. And that is, the production processes can be generic, and it produces consistently high quality, high purity RNA in just a matter of hours. Importantly, it seems to be quite a generic platform as well, as far as the product characterization and release. Typically, vaccines like recombinant protein vaccines require the development de novo of a wide variety of product-specific assays, such as their appearance, the impurities, identity, purity, content, potency, etc., all need to be developed de novo for every new product. This isn't the case for genetic vaccines, mRNA in particular, where most, if not all of these, might be able to be developed as a generic method that can be used from one product to the next. And this should allow us to radically simplify and accelerate the time it takes to develop a vaccine, going away from this linear approach that usually takes decades to a more concurrent approach where we can apply the genetic medicine technology to facilitate discovery and development, and therefore get the vaccines to those who need them much faster. The final issue about being ill-prepared to respond to emerging infectious diseases, I think a good recent example of how we fail on this front is the H1N1 influenza outbreak from 2009, where despite a very coordinated, massive effort between the vaccine manufacturers, governments, the WHO, the CDC, to accelerate the production of the influenza vaccine back in 2009. By the time we really had meaningful numbers of doses, the disease had come and gone. So clearly we needed to throw out the rule book when it comes to responding. We did have the opportunity back in 2013 to test a rapid response using the self-amplifying mRNA approach to the H7N9 influenza outbreak in China. And the sequence of that virus was posted on the internet on Easter Sunday in 2013. And because of an ongoing partnership that we had with the J. Craig Venter Institute, we quickly had them make us the synthetic template encoding the SAM replicon, encoding the H7HA. And within 8 days, we had vaccine in a vial ready to be used for immunization. In retrospect, if we had been truly ready and had all the reagents we needed and eliminated the 2 steps of mailing things from one location to the other and co-located all of the activities, we should have been able to do that in about 4 or 5 days. And in the bottom here, you can see the purified product, the HA that's expressed from the replicon, and the high levels of neutralizing antibodies that were elicited within 2 weeks after a single immunization in mice. So in principle, one can go from sequence to protective immunity in just a matter of weeks. So we envisioned at the time that a rapid response platform could look something like this, where an outbreak is declared, the pathogen is isolated and sequenced, and that information can be electronically sent to the locations where the vaccines are going to be manufactured. The initial step is to design the vaccine, which can be done completely in silico. In the case of an influenza pandemic outbreak, you could quickly hone in onto the hemagglutinin molecule and start production immediately. Of course, if it's a completely new pathogen never seen before, then some discovery work will have to be done to identify the target. But as soon as that sequence is available, then synthetic production can begin and very quickly have vaccine in a vial ready to distribute. And this is precisely the approach that was taken recently for COVID-19 by Moderna and their collaborators at the Vaccine Research Center at the NIH, where as soon as the sequence of the organism was made available, they quickly focused in on the spike-like-a-protein, engineered that spike-like-a-protein to be more stably expressed. And then within 45 days of the publication of that sequence, they had manufactured clinical trial material and began their phase 1 trial about 21 days later. And as we've all heard recently, last week, they announced very high levels of efficacy in their preliminary read, greater than 95% efficacy. So just in a matter of months, you can use this technology to enable a true rapid response to an outbreak of infection. Quite impressive. The landscape of COVID-19 vaccines is both deep and broad. As of last week, according to the WHO, there were more than 200 distinct programs, including 97 which are based on genetic medicines. Almost half of the preclinical programs are based on DNA, RNA, or viral vectors, and exactly half of the clinical stage programs are based on genetic vaccines. We also have protein-based approaches, inactivated virus approaches, and even live attenuated virus approaches. But the key front runners are DNA, RNA, and vectors, owing to the nature of the way the vaccines can be produced, released, characterized, and gotten to patients faster than the conventional approaches of protein inactivated viruses and live attenuated viruses. The landscape of mRNA vaccines includes all 3 different types of the RNA that I mentioned. CureVac is spearheading the approach of using unmodified bases of RNA, and they recently published some of their preliminary safety and immunogenicity data from their phase 1 trials and have ongoing trials in Europe. The base modified mRNA approach is being spearheaded by Moderna and BioNTech Pfizer, and they are both in late stage clinical trials, both in phase 3, have published some of their preliminary safety and immunogenicity data. And both of them have recently announced their preliminary efficacy data, with Moderna and BioNTech Pfizer showing about 95% efficacy, which is quite remarkable and a very strong evidence of the proof of principle that this type of technology can be both safe and effective in humans. The self-amplifying RNA approaches are being driven by primarily Imperial College, and we'll hear more about that from Robin Shattuck a little later, as well as Arcturus, both of which have phase 1 trials ongoing. And just last week, Arcturus announced that they have immunogenicity and safety data from their phase 1 clinical trial. So very exciting times for the technology, and to see that this might be a solution to the COVID-19 outbreak is quite heartwarming. So in summary, genetic medicines have the potential to really intervene at various aspects of the vaccines R&D process, including discovery by streamlining the antigen identification and optimization process. Typically with a recombinant protein-based approach, you're limited in the number of candidates that you can test because of the way that the vaccines have to be produced. But even if you're going to use a protein-based vaccine, you could still use the genetic approach of either DNA or RNA to very simply, with high throughput, express and characterize a wide variety of different antigen targets. Because genetic medicines have the ability to mimic a live virus infection and produce the antigen in situ, they induce potent and broad immune responses akin to what live attenuated viruses can do, but in a safer way. It appears that the technology is truly plug and play and can efficiently deliver a wide variety of different gene targets, at least infectious disease targets, including viruses, bacteria, and parasites, and in some cases also adjuvants. Because of the way the vaccines are made, using synthetic approaches, eliminating biologics altogether, using generic methods to produce, purify, and characterize the product, this should significantly simplify and reduce R&D timelines and really enable a rapid response against outbreak infections. And while I've focused mostly on vaccines and infectious diseases, this technology is also widely being used for both gene and cell therapy approaches, but also gene editing, where mRNA is being used to deliver the CRISPR-Cas9 to overcome some of the limitations that the DNA and viral vector approaches are encountering. So with that, I thank you very much for your attention, and I'd be happy to answer any questions.
Thank you, Jeffrey. We do have one question coming through. It says, what is your perspective as to why this capability was not further developed on the onset of COVID-19 pandemic? What is the role of government, industry, organizations like CEPI? What targets will industry prioritize?
Did I hear you correctly to say why wasn't this technology used for COVID-19?
That's what the question says.
Well, indeed it was. In fact, the front runners are genetic nucleic acid-based vaccines, in large part because of the technology being quite amenable to a rapid response. They could quite accelerate the time it takes going from the sequence of the gene of interest to having a vaccine ready to test. So indeed, I think, and just the sheer depth of the number of programs, both preclinical and clinical, I think is an attest to the fact that this technology, albeit quite new and untested, was used and is being used by a wide variety of companies, and to some apparent success.
Okay, thank you.
And I think for the second part of the question about the role of governments here, and what we've seen is that the governments have played a major role and will continue to do so. They've invested a large amount of money to de-risk some of the programs here for normally one wouldn't be doing things so much in parallel rather than waiting until we have some feedback and evidence that the vaccines are safe and effective before really launching into the large scale industrialization. But what the government funding has enabled us to do here is to do that in parallel so that we're ready to really have vaccines available faster than typical.
Okay, I think we may have time for one last question. There's a question as to whether mRNA, I guess the base modified mRNA and SAM approaches can work together in the marketplace, and if there's room for that.
Well, I think there is, in principle, at least the opportunity. I think if I understand the question correctly, can you use base modified form of a self-amplifying RNA? And I think there is an opportunity there to do so. And I think there has been some effort to see if that's possible. I haven't yet heard of any successes, but it in principle should be possible to have at least a portion of the self-amplifying mRNA molecules with base modified. But of course, once the replicon gets into a cell and starts amplifying, it's going to be making replicons with native bases.
Yeah, I think, sorry, I think the question was more about having two types of mRNA vaccines on the market at the same time, if there's room for that. And
I certainly think so. I mean, as I said, having the toolbox with the wide variety of different genetic medicines, not only RNA, but also possibly DNA and viral vectors. And there may be some instances where certain targets are better suited with one technology versus another. I think the one key potential advantage of the self-amplifying RNA is that it might achieve the same levels of protective immunity at lower doses. And there may be some instances where a lower dose of RNA is preferred to a higher dose of RNA. So I think that there's certainly room for not only both the base modified and self-amplifying, but some of the other genetic medicine technologies as well.
Okay, thank you. I think, Jane, correct me if I'm wrong, that may be the time we have for questions. Okay, go ahead, Andy.
Thanks, Shell, and thank you, Jeff, for a wonderful talk. So now I'd like to introduce you to our second speaker. This is Norbert Pardy. He's Research Assistant Professor of Medicine at the University of Pennsylvania, PhD in biochemistry and genetics, and he's a pioneer in the field of base modified mRNA vaccines. This is a genetic medicine symposium. University of Pennsylvania is a powerhouse in that field. We've seen, Carl June's team pioneering work in CAR T-cell therapy, and we've seen, base modified mRNA vaccines coming out of some of the work from, Norbert Pardy, Katie Carrico, and Drew Wiseman. So with that, I'll hand you over to Norbert, and he can give you a journey through some of his research endeavors.
Thank you very much for the kind introduction and the invitation. It's really my great pleasure to give a talk today. So as Andy mentioned, I'm going to tell you a bit about my SARS-CoV-2 vaccine studies, but I think for most of you, the first part of the talk will be a bit more interesting because I'm going to tell you about the short history of nucleoside modified mRNA therapeutics and highlight the most critical experiments and discoveries that led to the birth of this new class of drug. The most important reason why I really want to talk about this is because these groundbreaking experiments were performed here at Penn by two really fantastic scientists, Katie Carrico and Drew Wiseman. And as you all know, nucleoside modified mRNA vaccines are among the most promising vaccine candidates against SARS-CoV-2, currently being evaluated in phase 3 clinical trials by Moderna and Pfizer-BioNTech. So the first successful in vivo demonstration of the therapeutic potential of messenger RNA, in vitro transcribed messenger RNA, occurred in 1990 when Wolf and colleagues demonstrated protein production from in vitro transcribed messenger RNA after injection of mice, and despite the critical findings of this beautiful science paper, most people remain skeptical about the therapeutic potential of messenger RNA, and the field mainly focused on DNA-based approaches. The major issues were the unstable nature of messenger RNA, at least compared to DNA. It's less stable, so we had to figure out ways to make it more stable, and of course, in most cases, we use some kind of a delivery material that could protect RNA from extracellular RNases. A second issue was the really high innate inflammatory capacity of messenger RNA. So we have many different RNA sensors in different cellular compartments that sense foreign RNA, and we can easily induce a cytokine storm by the injection of normal RNA. And the third issue was the low in vivo translatability of the RNA. So we had to figure out ways how to produce more protein from a small amount of messenger RNA in vivo to see a therapeutic effect. But due to the technological advancements and very active research in the past 15 to 20 years, most of these issues were resolved, and messenger RNA became a really promising new therapeutic modality. So this figure shows, I think, one of the most interesting and most thought-provoking experiments that was performed by Drew and Coty 16 or 17 years ago. They published their groundbreaking immunity paper in 2005. So what they did in this study, they transfected monocyte-derived dendritic cells with different types of RNAs, RNA from mammalian cells, RNA from prokaryotic cells, and most importantly, in vitro transcribed messenger RNA shown in the purple bar, and they just looked at, they measured immune activation, TNF-alpha production by these different types of RNAs. And as you can appreciate, the conclusion was that, so they found that some RNAs were really immunogenic, particularly the in vitro transcribed, unmodified in vitro transcribed messenger RNA, and some RNA, for example, mammalian transfer RNA, was not very much immunogenic. And the conclusion was that natural RNAs are not equally potent activators of the immune system. And the question was, why this is happening? What is the difference between different types of RNAs? All RNA have the four building blocks of the four basic nucleotides. So Coty and Drew thought that maybe the difference is the different types of modifications. I just put one modification, the most abundant modification, pseudouridylation. I just put it here. So pseudouridine is made from uridine by pseudouridine synthase. It's really abundant. You can find it in ribosomal RNA, in transfer RNA, in both prokaryotic and eukaryotic cells. If you look at the percentages of nucleoside modification in various types of RNA and the immune stimulatory capacity of these RNAs, it became pretty obvious for Coty and Drew that most probably nucleoside, different RNAs containing modified nucleosides are probably less immunogenic. So transfer RNA in mammals that contains almost 25% modified nucleosides did not induce TNF-alpha production, but for example, in vitro transcribed RNA that was unmodified induced lots of TNF-alpha. To directly test this, they repeated the first experiment. They transfected monocyte-derived dendritic cells, but this time they used not only unmodified RNA, but also nucleoside-modified RNAs. It turned out that the incorporation of modified nucleosides can decrease immune activation. As you can appreciate in this figure, the unmodified RNA still induced a lot of TNF and IL-12, but it was significantly decreased after the introduction of modified nucleosides. So this was a very important discovery. A couple of years later, they found that the incorporation of modified nucleosides, particularly pseudouridine, not only decreases immune activation, but it also increases protein production from the RNA, as illustrated on as shown on this figure. So they transfected cells, both cell lines and murine dendritic cells, primary cells, with the vanilla luciferase encoding RNA, and then they just measured protein production by measuring luminescence, and they found that again the incorporation of pseudouridine or 5-methyl-C also increased protein production compared to the unmodified RNA. So with the nucleoside modification, you can kill two birds with one stone. You can increase protein production and you can decrease immune activation, and these are both very important. For many different therapeutic applications. So if we go back to this figure, you can appreciate that if you introduce modified nucleosides, you can significantly decrease immune activation, but there is still some residual immunogenicity. So Coty and Drew wanted to figure out what causes this residual immunogenicity and how is it possible to get rid of this. So it's known that the RNA polymerase that we use to produce in vitro transcribed RNA, it produces not only the RNA of interest, but also some byproducts. And Coty and Drew believe that most probably there is a significant amount of double-stranded RNA contamination present in the in vitro transcribed messenger RNA preparations. And it's also well known that there are many different double-stranded RNA sensors in the cytoplasm. I just listed some of them. And for example, PKR, protein kinase R, when it binds double-stranded RNA, it can phosphorylates eukaryotic initiation factor 2 alpha that shuts down translation. So obviously this is something that you don't want. So they tested this hypothesis and they used both unmodified and modified RNA. They performed a dot blot experiment with an antibody called J2 that recognizes double-stranded RNA. And indeed, it turned out that regardless of nucleoside modification, you always have a high amount of double-stranded RNA contamination in your preparations. So they figured out an HPLC purification procedure to get rid of these contaminations. This beautiful model was performed by Coty. So basically, it shows that if you HPLC purify the in vitro transcribed messenger RNA product, you still have a decent amount of immunogenicity if you use unmodified RNA, because uridine is recognized, published that uridine is recognized by toll-like receptor 7 and 8. But if you use nucleoside modification, for example, pseudouridine incorporation, you can get rid of this residual immunogenicity from your preps. So this, by around 2010, 2011, Drew and Coty developed really the therapeutic quality RNA, and this was the time when I joined them and started to work on solving other issues. And I always emphasize that you can use in vitro transcribed messenger RNA not only for translational research, but also to trying to understand and answer fundamental questions in multiple fields, for example, in the field of immunology, but you can use it for very simple experiments, for example, just a cell transfection study, because you can see that it works much better than plasmid DNA transfection. The transfection efficiency is typically really high, and you can transfect almost every kind of cell types. And if you transfect in the morning, you can collect your cells in the afternoon. But of course, we are very much interested in the translational aspect of nucleoside modified RNA. And we were, at that time, the biggest issue was that we didn't really have a good carrier molecule for messenger RNA. The problem was that in vitro mRNA transfection reagents are either toxic or they just don't work very well in vivo. So we were desperately looking for something that we could use to formulate the RNA and then use it in vivo for various purposes. And after, I think, 3 years or something like that, we bumped into Acuitas Therapeutics that produces a really nice special lipid nanoparticles. Peter will tell you more about them. And these were developed originally for siRNA delivery. And we just asked them to incorporate some firefly luciferase encoding mRNA into these LNPs. I just wanted to provide proof of concept that we can use these LNPs for mRNA delivery. And I used in vivo imaging for this purpose. I just immunized mice in many different ways and then looked at protein production over time. And here's the result. And the take home is that these LNPs are excellent mRNA carriers. If you inject animals systemically, IV or IP, you can produce tons of protein in the liver for a relatively short amount of time. But if you use ID or IM delivery or even sub-Q, you can produce protein for a longer time and many times in the injection site. And this is the favorable kinetics for vaccine studies. And this was our major interest. So this is the optimized nucleoside modified mRNA LNP platform or mRNA LNP vaccine platform that we use, and Moderna and Pfizer BioNTech uses pretty much the same technology. Of course, we don't know all the details about vaccine production, but again, the basic technology is the same. So we use a plasmid DNA template to produce RNA. The plasmid encodes some special 5' and 3' UTRs, ideal size of poly-A tail. We use a co-transcriptional capping reaction and to add the cap1 structure to the 5' end of the RNA. So we can do these 2 steps in basically one and in a single reaction to make the IVT RNA and capping. After this reaction, the RNA undergoes stringent purification. We switched from HPLC to cellulose purification. As a matter of fact, this cellulose purification procedure was developed by Katie Carico. This is much more scalable than the HPLC. So this was, and it's much simpler. So this is why we switched, and it works equally well. And this is important because we want to get rid of double-stranded RNA and all other contaminations that would otherwise be recognized as pathogen-associated molecular patterns by the immune system and induce unwanted immune responses. And of course, we replace uridines with 1-methyl pseudouridine. Like I mentioned to you, it increases protein production from RNA and decreases immune activation or inflammatory responses. So once we make this RNA, Acuitas Therapeutics formulates it into lipid nanoparticles. And again, this is what we use in our studies. So let me tell you briefly about my SARS-CoV-2 mouse vaccine studies. So it's known, so these are all known that SARS-CoV-2 infection, COVID-19 pandemic causes significant morbidity, mortality, and economical loss worldwide. Unfortunately, we don't have any vaccines against any coronaviruses, I mean human vaccines. But we could certainly learn from previous studies with SARS-CoV-1 and MERS. This was actually good news in January because we learned that the spike glycoprotein on the surface of the virus is a good vaccine target. And particularly the receptor binding domain of spike is a really good target. It contains multiple neutralizing epitopes. It was also good news, again, learning from previous studies that we knew that most probably we would be able to produce a protective vaccine against SARS-CoV-2 because we obtained similar results with SARS-CoV-1 and MERS previously. So, and of course, we would need a vaccine that induced durable responses, protective responses from SARS-CoV-2 infection. So, naturally, many academic labs and players in the industry jumped on it, and everybody started to develop vaccines using pretty much everything that we could use, genetic vaccines, and of course the traditional methods, adjuvanted protein subunit vaccines, inactivated virus vaccines, and live attenuated virus vaccines. And Jeffrey already showed these numbers that there are more than 160 preclinical candidates, roughly 50 clinical candidates, and most importantly, the nucleoside modified mRNA LNP vaccine platform is currently being evaluated in phase 3 clinical trials by Moderna and Pfizer BioNTech, and the results are very promising so far. So in January, I just decided to do a very quick proof of concept study, very quickly develop SARS-CoV-2 vaccines, mRNA LNP vaccines, and show that these are really effective. So I designed 2 different constructs, full-length S encoding mRNA with a furin cleavage site mutation. This was shown for influenza HA and many other antigens that if you mutate the proteolytic cleavage site, you will probably get a better immunogen, maybe it stabilizes the antigen. And the second construct was an RNA that only encoded the receptor binding domain of spike. And the experimental setup was very, very simple. We immunized biopsy mice with a single intramuscular immunization with 30 micrograms of mRNA LNPs. This is a very high dose, I know, but at that time in January, I didn't really know how great the vaccine will be, and I really wanted to go with a single immunization. It turned out that 3 micrograms would have been enough, but again, at that time it was not obvious, and I really wanted to come up with a protective vaccine. So basically, animals received a single immunization, and then we looked at the T-cell responses, B-cell responses, antibody responses, protective efficacy in animals, and we could publish this story in Immunity like 3 or 4 months after we started this project. So again, I think we could provide proof of concept. So in the first set of experiments, we just looked at T-cell responses. Animals received a single immunization again, and 10 days later, we collected spleen and lungs and looked at CD8 and CD4 positive T-cell responses, and we could measure pretty strong T-cell responses, both CD4 and CD8. CD8 positive T-cell responses were particularly strong in the lungs. We don't know exactly what it means, but we really want to look at it because, again, these responses were really, really nice with both vaccines. As we experienced previously, the Nucleoside Modified mRNA LNP vaccine very quickly induced high antigen-specific IgG titers as measured by ELISA, and titers were week 9 titers were higher than week 4. Again, this is a single immunization, and we always see these really nice durable responses in mice. And these antibodies had really high quality as measured by a pseudovirus-based neutralization assay. Titers at week 9 were significantly higher than titers at week 4. What we couldn't do and couldn't include in this publication is the protective efficacy study, because in March and April, when we perform these experiments, there was no appropriate mouse model for challenge studies. You cannot infect white type mice with a white type virus, so you need to figure out, and there are a couple of different ways to do these challenge experiments in mice. So what we did, we 5 days prior to the challenge, we injected mice with an adenovirus construct, human ACE2, and we could make mice susceptible to virus infection. So we challenged them 5 days after the injection of this At5 human ACE2. Again, we found that these mice induced really high RBD-specific IgG titers very quickly, 2 weeks post-immunization, and these were even higher 4 weeks, so just before the challenge. And we challenged them 20 days after the single immunization and measured viral titers in the lungs by plaque assay, and found that both the full-length furin mutant and the RBD vaccine induced sterilizing protection, no viral replication in the lungs of these animals. And you can appreciate that the control mice had really high virus titers. We looked at long-term, we looked at memory responses by measuring antigen-specific antibody-secreting cells in the bone marrow. So these are the long-lived plasma cell responses. Again, beautiful responses after a single immunization induced by both vaccine, and we got similarly strong memory B-cell responses. And I really hope that we will see something similar in humans, because memory is very important. So I will get back to this in a minute. But in mice, it worked. The vaccines worked really well. So if I just want to conclude these studies, we found that single immunization of SARS-CoV-2 mRNA vaccines induced high titers and of protective neutralizing antibodies. And at least in mice, these titers were really stable until week 9. That was the last time point examined. we found that the T cell responses were biased toward the Th1 profile, and we could measure really strong memory responses, so long-lived plasma cell and memory B cell responses 9 weeks post-immunization. So before I finish up my talk, just let me very briefly tell you about a couple of current limitations and questions about mRNA vaccines. So the good news that it seems that preclinical findings, both mouse and findings in non-human primates, translate to humans based on the studies by Moderna and Pfizer-BioNTech. In the initial, so it seems that this is going to be a two-dose vaccine. Companies use different doses. In the phase 3 trial, Moderna uses 100 microgram, BioNTech uses 30 microgram. In the first phase 1 trial, there, again, with higher doses, there were some adverse events, not terrible, but there were some, and I think particularly with the very high dose, 250 that Moderna used, and I think this was one of the reasons why they decided to use a lower dose, 100 microgram. Of course, there are many different ways to address this issue. We can play around with the doses. We can try different lipid nanoparticles. Moderna published a study on this. They tried different LNPs, and it turned out that they have different safety profile. And of course, we can develop other carrier molecules for the RNA. I don't think that the production is a problem for mRNA vaccines. Jeffrey also mentioned this. It's very simple, scalable. So I don't see any issues with that. We need to be careful with the phase 1 data because that was a very limited amount of people, like 50 people. And those studies didn't look at different age groups, sexes, ethnic groups at large numbers. Now we know that it seems that both vaccines, the Moderna and the Pfizer-BioNTech vaccine, work well in older people, but of course we need to see more data. The durability is also a big question. However, and it's known that SARS and MERS specific antibodies wane over time in people, and it's still a question what happens is after SARS-CoV-2 infection or immunization with SARS-CoV-2 vaccines. I don't think, as long as we have memory in people, I think it's a normal immunological response that there is a decrease in the antibody titers. You cannot maintain super high antibody titers forever, but if we have memory, we can boost these immune responses easier. It's also a question what happens if a new virus shows up. So there is a new dominant virus that carries a mutation called G614. It seems that this particular virus is actually even more susceptible to virus neutralization. So currently, I don't see any issues with the virus, with the mutation and the mutations, and the coronavirus SARS-CoV-2 doesn't mutate that fast, at least compared to flu. So we'll see how it goes, but right now it's not a problem. And let me just mention the very last aspect. So this is the storage. This is not completely correct because Moderna shared some information this Monday, just the day before yesterday, and they claim that their vaccine can be stored at 4 degrees for 30 days, and that's pretty good. I'm not sure if BioNTech really looked at the stability of their vaccine. Currently they say that it has to be stored at -70 degrees, and this needs to be improved. So finally, thank you very much for your attention, and I'm happy to take questions.
Great, thank you, Norbert. There was an initial question about, to clarify something, for the SARS-CoV-2 study. The question was whether that mRNA was a pseudo-uridine modified or a different.
Yes, it was. Yes, it was a cellulose purified 1-methyl pseudo-uridine modified RNA.
Okay, great.
It was, to my best knowledge, it was very similar to the Pfizer, BioNTech, and Moderna. Again, I don't know what exactly they use because companies don't share this information, but it must be very similar. To my best knowledge, they also use 1-methyl pseudo-uridine modification. I don't know what kind of purification procedure they use.
Okay, great. Thank you, Norbert. And with that, I'll hand it back to Andy.
Thank you.
So thank you, Norbert, for sharing your research. For the final speaker for this session, it's a pleasure for me to introduce Peter Cullis. He's Professor of Biochemistry and molecular biology at the University of British Columbia. He's a pioneer in the field of LNP delivery. I first came across his work when I was at Novartis Vaccines, where we were looking for an efficient way to deliver our self-amplifying RNA. The research was beautifully laid out, synthesis of those ionizable cationic lipids, the ethanol dilution process, a scalable process to produce the particles. We published a wonderful paper in 2012 showing LNP delivery of our self-amplifying RNA vaccines in PNAS. And it's thanks to Peter and his hard work that we were able to do that. I first met Peter because I was a principal investigator on a DARPA contract on RNA vaccines and their industrialization. And we'd organized a big delivery conference, and I was able to invite him to come and talk. And that was around 2012. That's the first time I've met him. So a pleasure to introduce you, Peter. Give us a tour of your research and lipid nanoparticles. Thank you. They've certainly changed the world. You're on mute, Peter.
Can you hear me okay now?
Yeah, we can hear you. It's great.
Okay, great. Thanks very much for the introduction, Andy. I'm going to go through the, now what's happened? Am I not sharing anymore?
No, you're not sharing anymore.
Right now, Peter, sorry about that.
Okay, I think that wasn't me. Something else happened. I don't know what it was.
There you go. You're perfect now.
Yeah, I just want to get on to the slideshow. Here we are. Is that all right?
Yep, you're great.
Okay, terrific. So today I'm just going to go through a little bit of background here as to how we got to where we are now. This has really been a lot of effort over many years for many people. And so I thought I would take you through some of those developments. I have a couple of conflicts of interest, Precision and Acuitas, both of which I'm a co-founder. Okay, so I'm going to talk a bit about the LNP systems for enabling gene therapies and emphasizing that it's been a 25-year effort. First of all, the Patisiran, or the Onpattro, the trade name now, story, which is a development of a drug to treat a disease you've all heard about. It's a relatively rare disease, hereditary amyloid transthyretin amyloidosis. This involved developing a system that allowed us to silence a gene in the liver. I'll go a little bit about. I'll spend quite a bit of time on the LNP program itself to achieve gene silencing in the liver. A little bit about the disease and some indication of the clinical results. Then I'll go on very briefly, and Norbert has given a very good introduction to this, to the development of an mRNA-based lipid nanoparticle drug as a vaccine. So the challenge that we came upon in 1995 was to design a non-viral system to deliver nucleic acid-based drugs. And of course, we started off with antisense. siRNA wasn't really known at that time, or RNA interference. So to deliver those drugs to the liver and enable intercellular delivery into hepatocytes. And as you're well aware, this is not a straightforward process. Nature has evolved many mechanisms to avoid nucleic acid polymers or nucleic acids in any form getting to the interior of a cell. And so I find it quite amazing that we've actually made some progress in this area. Just a word or two about the materials that we should use for these systems. And I've always taken the view that if nature does something, it's probably a good idea to copy it. And so nature uses lipids for delivery, and this is, lipoproteins, intercellular transport vesicles, exosomes, viruses, etc. And this is indicated here for influenza virus and LDL, the low density lipoprotein. One where you have an interior, we have lipid bilayer with the interior aqueous region, and the other where you have a hydrophobic center. Anyway, so there's a real message here. If nature is using this approach, it's probably a good idea to follow it. And over the years, we developed lipid nanoparticles that are increasingly good analogs of these biological systems. There's lots of advantages. So there's two major types, as you see on the left here. The bilayer systems that contain, say, an anti-cancer drug, and solid core systems where one can incorporate much larger molecules, DNA, RNA-based drugs. So these are self-assembling biocompatible systems. You're using lipids that occur naturally in nature, with the exception of the cationic lipids. There's very straightforward ways of making and loading these systems. As I'll illustrate, highly flexible. We can take advantage of natural targeting processes. And they're certainly by far the most clinically advanced of any nanoparticle carrier. There's now 11 lipid nanoparticle drugs that have been approved by the FDA, the European Medical Agency, and others. They're indicated here. Most of these consist of nanoparticles, bilayer systems with cancer drugs inside them. The one exception is Onpattro, which was approved in 2018, which contains siRNA, and I'll be focused mainly on that one for the next part of the talk. So by 1995, we had developed 3 of these formulations. These are formulations of anti-cancer drugs to reduce the toxicity and enhance the efficacy, so avoid sensitive organs and get more, to say, tumor tissue was the objective here. And these drugs do that to some extent, so you get a better therapeutic index out of these formulations. So we then said, okay, well, let's take on the challenge of using this sort of technology to enable genetic drugs. And we'd really, over the years, developed a fairly stringent set of rules for nanoparticle formulations of small molecule drugs. So these consist of drug trafficking efficiencies approaching 100%, low surface charge to reduce toxicity and also avoid rapid clearance. Long circulation lifetimes are essential, of course, if you're going to access a broad range of tissues. The formulation process, rapid, reproducible, scalable, small size, stable on storage, and optimized payout, drug retention and payout properties. And so we went right back to basics. And this was really before we did much work in vivo. How could we do this before nucleic acid polymers? So we chose the liver as our target tissue. And the objective between 95 and 2012 was to develop these systems to silence genes in the liver, in hepatocytes, as I said, initially with antisense and subsequently with siRNA. The liver is an obvious organ to attempt to develop these systems because there's a great variety of disorders that can potentially be treated by gene therapeutics in this organ. So first of all, we said, okay, how can we encapsulate nucleic acid-based drugs such as siRNA in a lipid nanoparticle? And we were used to molecules like doxorubicin, molecular weight of 540 in that case. And so for siRNA with a molecular weight of 13,000 kilodaltons, or 13 kilodaltons, there's a completely different problem. And obviously, in order to get association of these nucleic acid polymers with the lipid nanoparticle, we had to use something that was positively charged, and using positively charged lipids made the most sense. However, at that time, all available cationic lipids were permanently positively charged, and these are really, as most people are aware, very toxic molecules. There are no positively charged lipids in nature. And when you introduce these positively charged lipids to a membrane, they combine with endogenous negatively charged lipids and completely disrupt the biological membranes, so accounting for their toxicity. So what we did was develop ionizable cationic lipids with a pKa in the region of 6.5. And so these have the property that they're neutral. It's a neutral pH. let's say positively charged at low pH, the pH 4, about the acidity of a lemon. And we found we could load nucleic acid polymers into these systems at a low pH, and the contents were retained on raising the pH to physiological values. And so this goes back to about 1998 or somewhere in there. Our first generation ionizable lipid, pretty simple, a primary amine, and a couple of acyl chains, but having this property that the pH 4 is positively charged, neutral pH relatively neutral. So then we had to say, okay, well, we can maybe perhaps formulate using these systems, but how can we encapsulate the drug in a reasonable, scalable process? And so we developed this rapid mixing ethanol dilution process where the lipids are dissolved in ethanol, the oligo in water, pH 4 is indicated here, a very rapid mixing process. And then the first thing that falls out, you can think of this conceptually, the first thing that's going to fall out of solution is the negatively charged nucleic acid surrounded by the positively charged ionizable cationic lipid. If you do this fast enough, you can trap it in these limit size systems, as indicated here, and you can see intuitively how it is the potential for getting relatively complete encapsulation. So then again, you've got that after this, we dialyze away the ethanol, and these systems, as I mentioned, maintain integrity, and you raise the pH to pH 4. And we developed microfluidic mixing technology to achieve this process, the very rapid mixing process that's required. 3-to-1 aqueous stream to ethanol stream. Lipid composition is indicated here, about 40% of the ionizable, 40 to 50% of the ionizable cationic lipid. The rest structural lipids, cholesterol, diacetyl, phosphatidylcholine, etc. And you inject this mixture through the mixer, and then, as I mentioned, dialyze away the residual ethanol and raise the pH. In order to expedite this, we formed Precision Nanosystems, which has gone on to develop gold standard, I think, processes for manufacturing genetic medicines with the nanoassembler product line. I think these are a new class of nanoparticles, with a hydrophobic core, ideally suited, as I mentioned, to encapsulation of large macromolecules, such as negatively charged anything really, such as RNA and DNA. You can go to gold and iron oxide and other nanoparticles if they're negatively charged. You can get very high encapsulation efficiency. They're stable, monodisperse, and reproducible. You can adjust the size in a sensible manner as well. Okay, it's all very well to make these systems, but are they of any use? Do they work? So you'll note that there's no target information on the exterior of these systems. They take advantage of, I won't go into this, but they take advantage of a natural targeting process. They adsorb apolipoprotein E, which triggers uptake into hepatocytes, and they're treated like chylomicron remnants. But the next stage was to measure the in vivo potency of these systems for silencing genes in hepatocytes. We used a factor 7 mouse model that was developed by Kenneth Kink at Alnylam Pharmaceuticals. This is just simply consists of a system where we encapsulate siRNA against factor 7 in lipid nanoparticles, inject intravenously, and then assay for factor 7 in the blood some 24 hours later. So a relatively high throughput approach. We could do 2 experiments a week, and to optimize the nanoparticle system. What we found was that the potency of these systems was very sensitive to the species of cationic lipid that we employed. This was the period between 2005 and 2012, and we synthesized over 300 variants, all of which look pretty similar from what you can see here, the primary amine, etc., but which have very, very different potency properties. And so this is indicated on this slide. The y-axis here is a log plot, and you can see that there's a real maximum around about pKa with a pKa for lipids with a pKa of about 6.4. Whereas if you move about, let's say, as little as 0.5 pH units away in the pKa, the potency can be reduced by a factor of 100 or more. Here we're defining the potency as what the inverse of the effective dose at which 50% of the factor 7 in the in the liver or in the circulation was the dose required to reduce the circulating levels of factor 7 by 50%. So this is was certainly something that was what's the word, unexpected, and is very likely due to optimizing these systems so that they can escape from the endosome more efficiently. But the bottom line was that we moved from systems that were operating very close to the maximum tolerated dose to systems where we were about 1,000-fold away. So the effective dose was 5 micrograms of sRNA per kilogram body weight versus the 10 milligrams that we started off at. We did this without increasing the toxicity. If anything, the toxicity or the tolerability was improved. So this was work that took place, as I mentioned, over 2005 to 2012, and was one of the most remarkable collaborations I've ever been part of. And so we identified at this point MC3 as a gold standard, and that lipid is indicated here. So the results from this program showed that we could, we had this property of being able to silence factor 7. Of course, by extension, we can silence any gene by just changing the sRNA to target that gene. So then the question became, what gene should we silence, and will this work clinically? And so, in collaboration with Alnylam, we chose to develop a lipid nanoparticle system to treat the HATTR disease that I mentioned at the outset. So just a word or two about this disease. TTR is a protein expressed in the liver. It transports retinal binding protein. If there are mutations in this protein, it can form fibrils in the blood. These fibrils deposit presumably everywhere, but they have particularly nasty ramifications in nervous tissue and in the heart. So there's neuropathy, cardiotoxicity that results. There's really no effective therapy, and it's usually fatal within 5 years of diagnosis. It's a really very nasty disease, as is illustrated here. Of course, the real problem, well, many problems, but one problem is that you probably have a relative who's had this disorder. So if for individuals who show signs of this when they're in their 30s, they know what's in store for them. So the hypothesis here is very simple, to use to silence this gene in the liver, reduce the using sRNA to reduce the number of circulating tetramers, to prevent deposition of these fibrils, and perhaps, if you're really lucky, to promote recovery and stabilization by reversing the flow of these fibrils back into the circulation. So potentially, this is true of gene therapies in general, potentially a very simple solution to what is fairly obviously a devastating disease. So the clinical development program that Alnylam embarked on here for the LNP TTR system started in 2012. There was a phase 1 trial for healthy volunteers, and then went on to a phase 3 trial that was completed in August of 2017, as is indicated here. Now, the phase 1 study, this is in healthy volunteers, as I mentioned, showed that at doses of above 0.15 mg per kilogram, there was an immediate and reasonably long-lasting reduction in transthyretin in the liver when you're using an sRNA to silence that gene. And this lasted, or the silencing or the reduction in the levels of TTR in the circulation lasted for approximately 3 weeks to a month. So as a result of the study, a dose of 0.3 mg per kg every 3 weeks was chosen for the subsequent clinical trials. Now, a word or two on the phase 3 study itself. This was the study here was compared with There's a randomization process, 225 patients that were incorporated into the trial, 148 getting the siRNA drug, and then 77 getting sterile saline. Primary endpoint was a neural impairment score with many secondary endpoints, quality of life, weakness, ability to walk, the body index, etc. This is a wasting disorder, as you saw. The results were spectacular. This is indicating over the 18-month trial studies, the time it took to do the trial. You can see here that the placebo group in the sterile saline, their neural impairment score got worse over time, whereas those on that were receiving the drug, if anything, they got better. So let's think about this from a hereditary disease, where you're not only stabilizing it, but if anything, improving the situation for these individuals. So the results of the clinical trial were reported in 2017. The primary endpoint, neuropathy score. I always refer to this p-value. Whoever calculated this p-value must have really enjoyed it. Essentially 1 over Avogadro's number. I think it's a pretty spectacular indication that this drug actually works. Similarly, for the secondary endpoints, quality of life, muscle strength, gait speed, and other indicators, all of these are seeing very marked improvements. So this led in 2018 to approval of this drug by the FDA, the first FDA approval of an siRNA-based gene therapy drug. And we would argue this is a pretty big deal because you're not only halting the progression of a disease, but actually reversing damage. And I think, as I mentioned at the beginning, towards the beginning of this presentation, the simplicity of the argument you can make here for a therapeutic is a part of this as well. So you have, when you get right to the core of the matter affecting the genetics of an organism, you can really make a huge difference. So moving on now to the vaccine story, I'll talk a little bit about the gene expression in the liver resulting from lipid nanoparticles containing mRNA, and then very briefly about the COVID vaccine results. So in 2012, we started to ask the question, and this was really through Acuitas Therapeutics. Does the LNP technology enable mRNA-based gene therapies? And so this is the same structure as we saw for the siRNA, packaging the mRNA, and then asking the question, first of all, assuming that, of course, we can silence genes in the liver, can we now express genes in the liver using mRNA as opposed to siRNA? So the first question here was, does the lipid nanoparticle technology that we developed for siRNA enable mRNA? This is a very reasonable question. I tried to do this to scale for the siRNA on the top here, but if I really did the scale, you couldn't see the dots. So the point being, of course, that mRNA is considerably bigger than siRNA. And so asking the question, does this ethanol dilution and rapid mixing approach allow efficient encapsulation? And do lipids such as MC3 enable mRNA to produce proteins? The answer to the formulation process is yes. The same process that we're employing for the siRNA works well for mRNA. The cryo-TEM, the lower right here, these systems are often can be indistinguishable from what you'd see with siRNA. But instead of having, say, 700 copies of siRNA per lipid nanoparticle, 50 or 60 nanometer lipid nanoparticle, now we might have only 2 or 3 copies of, say, a 2 kilobase mRNA. Now we optimize these systems using a luciferase model that has similarities to the factor 7 model, but in this case, we're injecting lipid nanoparticles containing luciferase mRNA, assaying for luciferase in the liver, and then going through the same optimization process as we did for the nanoparticles containing siRNA. And so this is work that's been done at Acuitas over the last 5 years or so, screened, actually it's more like 300 again now, to develop these third generation lipids, starting with MC3, and then looking at the relative luciferase expression in the liver. This work has shown that we can now use the liver as a bioreactor to produce any protein we want. If you want to win the Tour de France, here's one example with erythropoietin getting supraphysiological levels of EPO and subsequent changes in hematocrit, etc., resulting from introducing a lipid nanoparticle containing mRNA containing mRNA coding for erythropoietin. This experiment was done in pigs. And as Norbert indicated, you can have gene expression arising from many routes of administration. This is indicated here for work that was done in Drew Weissman's lab with Norbert as the lead author, showing that intramuscular or intradermally, getting reasonably long-lasting gene expression following administration of these lipid nanoparticle systems containing luciferase mRNA. So then the question, of course, becomes: does this enable lipid nanoparticle enable LNP mRNA vaccines? You already know the answer to this. I don't have to go through this slide because we've had really a great talk on the vaccine aspect of these and other vaccines. Just safe to say that this is an approach that also seems to induce a strong, in addition to the antibody response, a strong T cell memory response as well. So this is work done again with Drew and Norbert, looking first of all at protection against Zika virus, injecting intradermally with the Zika virus pre-membrane glycoprotein, mRNA coding for that, and doing this, in this case, intradermally, and then challenging subsequently with the Zika virus itself. And this just shows that at 2 weeks and at 20 weeks, there's complete protection against this challenge, as is indicated here. This, of course, led immediately to work that Acuitas is doing with BioNTech and with others, CureVac, obviously switching. This work started off as collaboration to develop cancer vaccines, but of course in January switching over to developing vaccines against COVID-19, SARS-CoV-2, and using, in this case, the glycoprotein approach. So this just indicates the mRNA that was employed. And as you're aware, there's numerous studies coming out now where this is found to be more than 95% effective, more than 90% effective for protection against SARS-CoV-2. This is data from Pfizer and BioNTech that was announced November the 9th. So we would expect that the application is going to be made to the FDA for emergency use probably by the end of this month. Similarly, Moderna's vaccine candidate announced similar results on November the 16th. This lipid nanoparticle has very similar composition to that which the Pfizer BioNTech vaccine has, but really pointing out that this approach, the mRNA approach in lipid nanoparticles, is working rather well. So it's been quite a journey. It's a 25-year effort, certainly going in very unexpected directions. And it's a collaboration with Norbert and with Drew Weissman at the University of Pennsylvania has been seminal in developing the vaccine aspect of this approach. So a very the paths that one takes in research certainly aren't exactly linear. Let's put it that way. But in this case, it worked out rather well. I think in the future, if you look to the future, the lipid nanoparticle approach, mRNA, using mRNA in particular, is really going to enable gene therapy in all its forms. We're certainly seeing that for the vaccines, and perhaps a universal flu vaccine and vaccines for cancer may be in the cards. We can use the liver as a bioreactor to produce proteins. We don't know yet whether the liver is going to have a problem with producing proteins that it doesn't normally, and we shall find out about that. Gene editing applications, extrahepatic tissues are now coming online following an IV injection, immune cells, bone marrow, et cetera, et cetera. And so there's many, many applications that one can see on the horizon. This is the work. This work has involved literally hundreds of people over the last 25 years. Some of them are indicated here. I particularly want to acknowledge Mick Hope, Tom Madden. When I started my lab in 1980, Mick and Tom joined me at that time, and we've worked together more or less continually for over 40 years. So I think it's these long-term collaborations that really can make a big difference. There's many others who have contributed hugely, and they're indicated here: Steve Ansel, Ying Tam, Barb Mui, and Paul Lin, Sean Semple, James Taylor, Ewan Ramsey, Lloyd Jess at Precision Nanosystems, and Carl Hansen. This is on the microfluidics side. Alnylam, a remarkable collaboration with Alnylam over the period mentioned 2005 to 2012. Marcus Ciflini, who really broke the problem of the ionizable lipids in the late 2008, 2009. These things work well in the brain as well. I haven't mentioned that. My own group in the biochemistry department, and of course, Drew Weissman and Norbert Party at the University of Pennsylvania. So I just leave you with this message. I think we're in for some very interesting times in terms of new therapeutics, not just in the vaccine area, but in many other applications. And with that, I'll take any questions.
Great, thank you, Peter. We actually have a couple questions about the structure of the lipids, the ionizable cationic lipids. So maybe I'll just sort of try to paraphrase and combine them together. But maybe if you could elaborate a bit on some of the trends as to how the structure of the lipids impart different favorable properties, like the length of the chains, degree of saturation.
Yeah, I'm not a chemist. I mean, all I know is the structures get uglier and uglier as time goes on. We started off with a simple precept. If you add the cationic lipid to an anionic lipid, as I mentioned, they tend to form these membranes disruptive hexagonal, this non-bilayer phases, and so we knew that this is a logical enough way to say escape from the endosome, and so we were designing them so that they had optimal bilayer destabilization process capabilities when they combine with negatively charged lipids. Whether that's a true rationale or not, I don't know. To this point, what's being done is phenomenological. Certainly the pKa, optimizing in that region, that seems to be a kind of a golden rule in these formulations. But the structure where one has 2, 3, 4 acyl chains, in some cases that's what we're seeing as people move to a more potent systems. The issue that you can face when you get these more complicated lipids is they may work well, but the formulation process and the ability to encapsulate mRNA can be affected, and so that's often where you end up, in a sense, jettisoning some of these candidates simply because you can't formulate, you can't encapsulate, even though they logically, from a pKa and other points of view, should be working well. I didn't give you a direct answer on that. It's, as I said, it's still phenomenological in terms of the optimization process. It'd be very nice to have an in vitro process that was a little more, a little easier to go through an optimization process as opposed to an animal model, which is what we're using currently.
Thank you, Peter. There are also a couple questions about sort of non-liver targets and different routes of administration, and maybe I'll try and agglomerate those together as well. But currently, these LNPs are IV injected and passively targeted to the liver. But are there other ways or other optimizations you could do for, say, subcutaneous injection or targeting to other organs of the body?
Yeah, the trick there is going to be, right now the systems that we're using for both for the vaccine and for the liver applications, if they're injected intravenously, they have very short circulation lifetimes. And so to get to other tissues, the initial mandate is that we have to have long circulating systems that are transfection potent, and that's been a bit of an issue to try and develop. I think we're on the verge of doing that, and probably the first tissue you're going to see that we can transfect with some reasonably high levels of potency is going to be bone marrow, where of course there's quite a few potential targets that are every tissue that you can hit, there's going to be more. We're going to face some issues that it's going to be difficult to avoid transfection in the liver, even though you're able to get more material to these other sites. So these are, these are issues that we will face going down the line, but you're certainly going to see that we're increasingly able to get systems that where we can do gene therapy at many sites in the body following an IV injection. And making that more and more specific is really the next phase of this game.
Okay, thank you, Peter. And with that, I'll throw it back to Andy.
Okay, thank you, Shell. So with that, I'm going to close the session. Just say thank you to 3 of our speakers, and wonderful tour through a lot of history. Thank you very much.